Welcome class to chapter 7. This is really our introduction into the nervous system. So we'll review a little bit of the anatomy of the nervous system, and then we'll get into some of the pathways of the nervous system. This PowerPoint is extremely long, and I'm going to go through it fairly quickly. So I would encourage you to watch maybe half of this lecture at one time and try to study that much, and then come back to the lecture and try to study the second half. So try to separate this chapter off into two chunks, but I will be going through it more quickly than I normally would if we were in person. So please just take your time, read the textbook. I also uploaded the answers to all the end of chapter questions for this module. So please take a look at those to make sure you're studying the right material and you're comprehending things. So a little bit of the introduction to the nervous system. This might be a review from anatomy, so I will go over this relatively quickly. We have your central and peripheral nervous system, and you should understand what makes up each. And then you should really understand what makes up the tissue and what are the two main types of cells, the neurons and the neuroglial cells. This is a great kind of summary of different terms in the nervous system, the definition, and then the differences between an interneuron, sensory neuron, motor neuron, the nerve itself, a somatic and autonomic nerve, the ganglion, nucleus, and tract. And you should have a really good understanding of these terms. Hopefully a review from anatomy, but please spend some time on those terms as well. The general functions of your neurons, which are the structural and functional units of the nervous system, are to respond to chemical and physical stimuli, conduct electrical impulses, release chemical regulators, and enable perception of sensory stimuli, learning, memory, and control of muscles and glands. That's really what this chapter is all about, is going through all of those functions of neurons. Most of them cannot divide, but they are able to repair themselves. So here's the general structure of a neuron. You should be familiar with the cell body, dendrites, and axon, and be able to label a picture like this to understand the different parts of the neuron. So here are the axons. They're connected to the cell body by the axon hillock. And the axon hillock is important because that's where the action potential will be generated, and we'll come back to that. They're covered in myelin with open spots called nodes of Ranvier. And an active process needed to move organelles and proteins from the cell body to axon terminals is known as axonal transport. It's a fast component which moves the membranous vesicles, and slow components will move microfilaments, microtubules, and proteins. So these are parts of a neuron, again showing the axon hillock. The nodes of Ranvier are where the action potential will kind of jump between these regions of myelination. And again, the myelination helps to speed up the conduction of the transmission of an action potential. It's kind of like electrical tape or insulation. So these are functional classification of neurons based on the direction of impulse. You should know the difference between a sensory and a motor neuron. Sensory conducts impulses to the CNS, your brain and spinal cord, and motor neurons conduct impulses from your CNS to on any target organ. Any interneuron is located completely within the CNS and will integrate functions of the nervous system. So this takes you through motor neurons. They can be somatic, which are responsible for all voluntary control of skeletal muscle, or autonomic, which will innervate involuntary targets. And this is where we get the sympathetic or parasympathetic divisions. They're sympathetic, functions in emergency situations, the fight or flight, and parasympathetic functions in normal, everyday functions like digestion, and that's our rest and digest division. So you should have a really good understanding of those two divisions of the autonomic nervous system. Then this is just a general look at the types of neurons involved, whether they're in the central nervous system or peripheral nervous system, showing the direction of impulse, whether it's a sensory or motor neuron, and then showing what a somatic motor neuron goes to and what an autonomic motor neuron goes to. Structural classification of neurons. They can be pseudo-unipolar, bipolar, or multipolar, and that has to do with the number of processes that are on each end. And this is a look at the different structural classification of neurons. More classification of nerves. Nerves are just bundles of axons put together located outside of the central nervous system. And you can read through some nerves contain both sensory and motor neurons and are mixed. Some of the cranial nerves have sensory fibers only, and a bundle of axons in your central nervous system is called a tract, and we'll come back to that. Neural glial cells, and these will be important to remember. There's many different types of neural glial cells. And again, I hope you guys had some of this in anatomy, because I'm hoping some of it's a review. If not, I'm sorry, I'll have a little more catch up to do. There's two types are found in the peripheral nervous system. Schwann cells, which are neural lemocytes, they will form myelin sheaths around the axons, and satellite cells support the cell bodies within the ganglia of the peripheral nervous system. Then there's 4 types of neuroglial cells found in the central nervous system. And again, backing up here, the 2 types of cells in the nervous system are neuroglial cells, or just glial cells, and your neurons themselves, which are made of the axon, dendrite, and cell body. So we're going over the neuroglial, or the support cells now, and you should have a good understanding of what the 4 neuroglial cells are in the central nervous system. The next few slides take you through the 4 types of neuroglial cells in the central nervous system, and this is a great chart to go over and review. I can guarantee I will ask you questions about these neuroglial cells and their functions on the next exam. So I'll let you work through what creates the myelin sheath in the peripheral nervous system. And you can see that there. Again, the myelin sheath surrounds the axons, helps to increase the speed of transmission of the electrical impulse. Here's a look at an unmyelinated versus a myelinated axon, and you can see there's a big difference here between the 2 axons. So there's some axons that are unmyelinated and some that are myelinated. More myelin sheath in the central nervous system is made by oligodendrocytes. And again, I'm just going to kind of pass over this. You should know why things are white and why things are gray. The myelin will be associated with tissues in the white matter, and gray matter will be associated with cell bodies and dendrites, which lack myelin. Demyelinating diseases. So this is a clinical significance of myelination, and it's important to understand these because multiple sclerosis can affect young women. The Guillain-Barré syndrome can also affect people, but multiple sclerosis might be a little more common that you'll hear about. So understand what goes into those 2 syndromes. Regeneration of a cut neuron. Again, neurons can regenerate themselves or regrow. And this explains that process going through these steps. If an axon is cut, it just shows how the axon will continue to degenerate first, and then growth and regenerate, reconnect it, and reestablish a connection to the skeletal muscle or gland that it innervates. So this takes you through some regeneration. Neurotropins promote neuronal growth in the fetal brain, so this has to do with development. In adults, neurotropins aid in the maintenance of sympathetic ganglia, which are clusters of cell bodies in the nervous system, and regeneration of sensory neurons. So now we get to glial cells, and I'm going to let you guys go through this, but you should have a good understanding of what astrocytes do, the blood-brain barrier, shown here. Astrocytes more astrocyte functions, and how they connect to neural activity, especially in the blood-brain barrier. So electrical activity in axons, this takes us into the sodium-potassium pump and how we establish action potentials. And I would encourage you to watch the video that I posted on YouTube about action potentials and how they're initiated, and then how they travel through the axon. Neurons have a resting membrane potential of -70 millivolts, and that's established by large negative molecules inside the cell, which is what we kind of went over in the previous chapter 6. Also, they're established by sodium-potassium pumps and how permeable the membrane is to positively charged ions. It's important to know that there's a higher concentration of potassium inside the cell and sodium outside the cell at rest of an axon. So what happens during an action potential is these ions will flow into or out of the cell to change the membrane potential of that membrane on the axon. At rest, a neuron is considered polarized when the inside is more negative. At rest, it's negative 70. When the membrane potential inside the cell increases and it becomes more positive, we call that depolarization. And a return back to resting potential is called repolarization. If the membrane potential inside the cell becomes even more negative than resting, we call that hyperpolarization. So changes in the membrane potential can be recorded in the axon by placing an oscilloscope, which has 2 electrodes, and they can record the voltage inside and outside the cell. And this is how we learned about how action potentials travel down an axon in a neuron. And an action potential is just the way the nervous system sends out its stimuli or sends out its responses. So depolarization will occur when sodium positively charged ions enter the cell, and hyperpolarization occurs when positively ions leave the cell, which are usually potassium. Depolarization of the cell means it's excitatory, meaning that is what when the signal is being passed down the neuron, and hyperpolarization is inhibitory, meaning when the axon of the neuron is in hyperpolarized state, another action potential cannot be generated, meaning another stimulus can't be sent down the axon. So this takes us through changes in membrane potential are controlled by changes in the flow of ions through different types of channels, whether they're sodium or potassium. Ion gating in axons, the voltage-gated sodium channels, these channels will open if the membrane potential will depolarize to negative 55 millivolts, and this is called the threshold. Sodium will rush in due to the electrochemical gradient. That means it'll go from an area of higher concentration of sodium outside the cell, and it'll rush into the cell. And membrane potential will climb toward the sodium equilibrium potential. These channels will be deactivated at positive 30 millivolts when an action potential is reached. So this is showing a closed sodium channel at resting, and the channels will be open by depolarization, meaning the action potential, the signal will be sent down the axon, and sodium will rush in, and then they'll be closed again during what we call a refractory period, a hyperpolarization state. Voltage-gated potassium channels at around positive 30 millivolts, voltage-gated potassium channels open, and potassium will rush out of the cell, and this is what we call repolarization. At the threshold membrane potential, which is negative 55, voltage-gated sodium channels open, sodium rushes in, the cell becomes depolarized, which causes more and more sodium channels to open, and this is called a positive feedback loop mechanism. This will happen until the membrane potential reaches positive 30 millivolts, and this is called depolarization. At positive 30 millivolts, sodium channels close, potassium channels open, and potassium leaves the cell. And this will result in repolarization. This will be a negative feedback loop as it tries to bring that axon back to resting potential. So I'm just going to focus your attention on this graph on the right. If a stimulus is applied to an axon, if it gets past this threshold, which is negative 55, which means if enough sodium rushes into the cell, we get past the threshold and we reach an all or none response. That means once we reach the inside of the cell to negative 55, sodium will continue to rush in until we reach an action potential at positive 30. At that point, sodium channels close, potassium channels open, and potassium rushes out of the cell. When positive ions rush out of the cell, the inside of the cell becomes more negative again. It will become back to resting and possibly even hyperpolarization, which is this little dip down here. So here are action potentials, what happens at each phase of depolarization, repolarization, and then hyperpolarization, and then which channels are open, inactivated, and open again. After hyperpolarization, repolarization will actually overshoot the resting potential, so we get our axon down to about negative 85. The sodium-potassium pump will quickly reestablish the resting potential, meaning if we're at negative 85, it'll pump sodium or potassium ions in and out of the cell to get the resting potential back up to negative 70. The all or none response law says that once that threshold of -55 is reached, an action potential will happen, and the size of the stimulus will not affect the size of the action potential after it reaches this all or none threshold. It'll always just reach +30. So here's a look at all similar amplitudes of action potential. And you can see here that even with a stronger stimulus and a stronger stimulus, the action potentials will have the same peak size, regardless of how strong the stimulus is. The stimulus just has to be strong enough to get the axon up to -55 threshold. So I'm going to let you look at these. This is coding for stimulus intensity. If we were in a lab, we could measure the intensity stimulus sustained for indicated periods of time. So these are action potential peaks showing how strong a stimulus is. You can see that this stimulus is not strong enough to generate an action potential, but once we get up to this stimulus, it is strong enough to generate an action potential. And if the stimulus is on for a longer amount of time, it'll just generate more action potentials. Refractory periods are talking about how action potentials can only increase in frequency to a certain point, and the absolutely refractory period will occur during the action potential when sodium channels will remain inactive. And this, when sodium channels are inactive, they will unable to be open again due to this refractory period. And the relative refractory period is when potassium channels are still open. So we have an absolute refractory period occurs when sodium channels will be inactive, and a relative refractory period is when potassium channels are still open, but only a strong stimulus can overcome that. So this shows the absolute refractory and relative refractory period. Again, the refractory periods are important, just meaning another action potential can't be generated in this exact same location in the axon until the refractory period is over. This is important so that the action potential travels down one direction in the axon, so that this helps in your nervous system for things to move as quickly as they can to get to where they're supposed to go, whether that's your central nervous system or some sort of gland or muscle. Cable properties of neurons. Neurons can conduct charges through their cytoplasm. I'm going to just kind of skip over these slides for now. I really want you to understand the action potential and really what channels open and close during that. More conduction of nerve impulses showing the direction of where the action potential will travel down. Conduction in an unmyelinated neuron. The conduction rate is slower in an unmyelinated neuron. So myelination just helps to increase conduction rates, and that's due to the nodes of Ranvier. The action potentials will leap from node to node, and we call that saltatory conduction. And that's really because your sodium channels are concentrated at those nodes, and we know that we need sodium channels to open up to depolarize the axon to actually get an action potential to occur. So here's a look at conduction in myelinated neuron due to saltatory conduction. Action potential is increased by the diameter of the neuron and also myelination. So action potential speeds will be greatest in larger diameter neurons and also if they're myelinated. And this just gives you examples of thin unmyelinated neuron speeds, and thick myelinated neuron speeds are much faster. You should know that as well, what speeds up action potential speed. All right, so here's a look of examples of functions served based on the diameter and the conduction velocity. So slower ones, slower velocities of action potentials will go to smooth and cardiac muscles. Much faster ones will go to sensory muscle positions, and all of basically your skeletal muscles will have extremely faster modes of conduction. And that's good because if you put your hand on a hot stove, you want your muscles to pull it off right away. So that's why your muscles are very fast. The synapse. This is a functional connection between the neuron and the cell it's signaling. In the central nervous system, the second cell will always be another neuron, but in the peripheral nervous system, the second cell will be a muscle or a gland. So we call it a myoneural or a neuromuscular junction. If one neuron is signaling another neuron, the first is called the presynaptic and the post is called the postsynaptic. And in between, there's a synapse or a space. There's different types of synapses. Most synapses are axodendritic, meaning they're connected from an axon to a dendrite of another neuron. And synapses can be electrical or chemical. This explains electrical synapses, where they're located, how cells are joined together, and what sort of stimulation will do to the connexin proteins to open or close the channels. Here's a structure of gap junctions. Again, gap junctions will connect two cells together, and it'll just be a way for things to pass through the connexin proteins. Most synapses involve the release of a chemical called a neurotransmitter. The synaptic cleft is small, and they're held together closely by cell adhesion molecules. And this is a really cool picture to look at, just the synaptic cleft, the synaptic vessels, and how they're kind of held together in the chemical synapses. The release of the neurotransmitter, it'll be enclosed in a synaptic vesicle, and it'll be released following these steps. Calcium and synaptic vesicles, when calcium enters the cell, it'll bind to a protein. Basically, the calcium will help the calcium will help the vesicle to be released. So this is a very good summary of the release of the neurotransmitter, and you should probably know these 4 steps. The action potential, which we understood goes through repolarization and depolarization through the sodium-potassium pump, sodium and potassium channels. The action potential will then reach the axon terminal where the neuron ends. It'll open up the voltage-gated calcium channels, which will allow calcium to enter into the area of the axon terminal. Calcium is important because it will bind to sensor proteins in the cytoplasm, and then this calcium protein complex will stimulate the fusion and exocytosis of the neurotransmitter. So calcium is another important ion needed in the whole nervous system process because it helps to release that neurotransmitter. Actions of the neurotransmitter, it'll diffuse or travel across the synapse where it will bind to a specific receptor protein. The neurotransmitter is referred to as the ligand, and this results in the opening of a chemically regulated ion channel, also called a ligand-gated channel. When this ligand-gated ion channel opens, the membrane potential will change, and it will change depending on what channel it's referring to. And I'm going to let you guys read through this. We don't have to know too much detail about these, but this is another good summary of neurotransmitter action on the presynaptic neuron and postsynaptic neuron. And again, the synapse is the space where the neurotransmitter gets released from. Understanding the role of calcium and then understanding what the neurotransmitter does. It'll open chemically ligand-gated channels, which will cause an inward diffusion of sodium, which will cause depolarization, localized decremental conduction of that depolarization, the EPSP. It'll open more sodium-gated channels and then potassium channels to eventually conduct the action potential down the next axon of that neuron. Acetylcholine then, this is one of the most important neurotransmitters. It directly opens ion channels when it binds to its receptor. Some cases, acetylcholine is excitatory, so it will excite the next neuron. Other cases, it will inhibit the next neuron depending on the organ involved. And you can see where it might be excitatory and where it might be inhibitory. There's 2 types of acetylcholine receptors. They can be nicotinic, which can be stimulated by nicotine, and they will be found in the motor end plate, we call it, of skeletal muscle cells. Or muscarinic can be stimulated by muscarine from the poisonous mushrooms. This will be found in your central nervous system and the plasma membrane of smooth and cardiac muscles. Agonists and antagonists will be drugs that can stimulate the receptor, nicotine for nicotinic acetylcholine receptors, and muscarine for muscarinic acetylcholine receptors. And antagonistic will be drugs that inhibit the receptors, atramine and curare. The binding of the neurotransmitter to a receptor can open an ion channel either through a ligand-gated channel or a G protein coupled channel. This takes you through the ligand-gated channel opening from the acetylcholine. And you can read that through here and also look at the nicotinic acetylcholine receptors. Ligand-gated channel opening, a little more detail there. Graded nature of these EPSPs. This is a comparison of EPSPs. The excitatory postsynaptic potential, again, is what the EPSPs stand for, and an action potential. Another word for EPSPs is just a graded potential versus an action potential. Knowing the general differences between the two is important. Basically, a graded potential has to do with neurotransmitters, and the stimulus for opening an action potential is depolarization. Localized are more graded, and the action potential will be regenerated over the entire axon. G-protein coupled channels will be when the neurotransmitter receptor is separate from the protein that serves as the ion channel. And I'll let you guys read through some of the details in the G-protein channels. I won't ask you too many specifics about these. Steps in activation and deactivation of G-proteins, I will let you read through this. G-protein coupled channels, again, we kind of talked about this a little bit in chapter 6. I know this really gets into a lot of details, but when we start talking about cardiac cycle, your SA node, you'll understand when we bring up G-protein coupled channels, kind of a little bit what we're referring to. More G-protein coupled channels continued. A lot of detail here. I won't ask you a lot of detail, but knowing in general what the two types of channels are. Acetylcholinesterase is important, often abbreviated ACHE. This will be an enzyme that inactivates acetylcholine activity right after it binds to a receptor. It basically cuts apart acetylcholine into acetate and choline, and that will be taken back into the presynaptic cell to be reused. So acetylcholinesterase inactivates acetylcholine, and you should know that. This is a very important enzyme to know. That this is because it'll control, it's just another controlling mechanism for a transmission of a stimulus throughout your nervous system. Acetylcholinesterase, it controls the amount of acetylcholine getting across the cleft. Cholinesterase inhibitors will be drugs that block the action of acetylcholinesterase, increasing the amount of acetylcholine in the synaptic cleft, and actually will enhance the synaptic transmission. So it's kind of like a double negative here. We have another enzyme inhibitor, but it's inhibiting another inhibitor. So cholinesterase will actually increase the amount of acetylcholine. These are 3 different molecules that treat myasthenia gravis and are important to help treat Alzheimer's. However, nerve gas and organophosphate pesticides can kill their intended victims by inhibiting acetylcholinesterase and overstimulating these synapses. So what actually nerve gas does, nerve gas is a type of a cholinesterase. If we inhibit this acetylcholinesterase, we're constantly flooding that synaptic cleft with acetylcholine. So we're just overstimulating our neurons, and that's what nerve gas will do. Somatic motor neurons will form interactions called neuromuscular junctions. Remember, a somatic neuron always will innervate a skeletal muscle. So these will be important to innervate skeletal muscle cells. If we interrupt the neuromuscular transmission, there's certain drugs that will block the neuromuscular transmission. Curare is an antagonist of acetylcholine, so it will block the receptors. So acetylcholine can't attach to those receptors, so muscles will not contract. So curare, and I'm not even that, there's a couple ways to pronounce that, this could lead to paralysis and death, and due to it paralyzing your diaphragm. If your diaphragm gets paralyzed, you'll be unable to breathe. So this is a major muscle that it will affect. It will be used clinically to relax muscles, but too much could lead to paralysis and death. So this is a great look at the drugs, the origin of the drug, so whether it was artificially made or coming from snakes like a toxin, and then the effect of it. So botulism toxin produced by a bacteria will actually inhibit the release of acetylcholine. That's what is used in Botox injections, so you probably never want to put that in your body. Nerve gas is an artificial, so it's man-made. It'll inhibit acetylcholinesterase. And then tetrodotoxin is actually involved in pufferfish, which some people eat as a delicacy. This will block sodium channels, which is also bad. So it surprises me greatly that some people do this to themselves, like Botox, or they'll eat a pufferfish. I would never get close to any of these drugs, but you can read through here and what the effects will be. Alzheimer's disease. This is associated with the loss of cholinergic neurons that will synapse on the areas of the brain that are responsible for memory. So that's what Alzheimer's disease does. It's a loss of neurons that will synapse in areas of memory in the brain. Myasthenia Gravis is an autoimmune disease. Whenever you see the word autoimmune, that means the body attacks itself. This is caused by antibodies that will block acetylcholine receptors. So this could also lead to muscle weakness or paralysis. Monoamines as neurotransmitters. So we really talked about acetylcholine. Monoamines can also be neurotransmitters. They're regulatory molecules derived from amino acids, catecholamines, serotonin, histamine. Monoamine action and inactivation are very much like acetylcholine. They'll be released, diffused across, bind to receptors. They'll be taken back up into the presynaptic cell, and they'll also be degraded by what we call monoamine oxidase, which is another enzyme that will degrade them to control them. So this is monoamine action and inactivation, very similar to the acetylcholine action and inactivation. Oxidase inhibitors, again, these will be drugs that block the degradation. So inhibitors will be proven useful in the treatment of depression, they found out, as well as panic disorder, anxiety, and Parkinson's. They can have potentially dangerous interactions with over-the-counter drugs that might contain tyramine, and such interactions could provoke a hypertensive crisis. This is why you want to have a really good pharmacist. So pharmacists are incredibly smart because they understand how all these drugs interact with each other. One drug might have a benefit, but if you're taking something else, it could have a bad interaction. Monoamine action, none of them use a direct ion channel. They all use a second messenger system, like the cyclic adenosine monophosphate, that cyclic AMP. Remember that second messenger that we talked about in the last chapter. So I'll let you read through how this cyclic AMP helps to activate an enzyme which will phosphorylate other proteins to eventually open a channel that began with this monoamine action. Norepinephrine action in G proteins, it will bind to a receptor using a G protein subunit dissociating binding. Also using cyclic AMP, so it kind of uses G proteins and cyclic AMP to eventually open up an ion channel. So you can read through norepinephrine. Serotonin is used by neurons in the brain. It's implicated in mood, behavior, appetite, and cerebral circulation. The drug LSD and other hallucinogens may be agonists to serotonin. So serotonin-specific reuptake inhibitors are used to treat depression, and this is what Prozac, Paxil, Zoloft are. They're basically they're inhibitors for serotonin reuptake, meaning they're leaving more serotonin in the brain to help keep you happy. Simply put, serotonin, over a dozen known receptors allow for a diversity of serotonin function. But again, different drugs that target specific serotonin receptors can be given for anxiety, appetite control, and migraine headaches. So, I mean, based on what the diagnosis is, you would get a different drug that will target a specific receptor that might have to do with anxiety, appetite control, migraine, depression. Dopamine, neurons that use dopamine are highly concentrated in the midbrain area of the brain, involved in motor control, emotional reward. That's dopamine. More about dopamine, I'm going to let you read through this on your own. I won't actually ask you a lot of, again, specifics about this either. But if you're interested in this, because a lot of these drugs are incredibly common, And many of us are on drugs like this. So if you're interested in it, learning more about it, I encourage you to really read the textbook. Norepinephrine is used in both central and peripheral nervous system. The sympathetic neurons use norepinephrine on smooth muscles, cardiac muscles, and glands. That's the fight or flight. So your norepinephrine is used in emergency type situations, and it's also used in the central nervous system. Other neurotransmitters, excitatory NT-glutamate, amino acid used to help excite. We'll keep going through some of these. Glutamate receptors, I'm going to let you guys read through some of these on your own because I know we're getting kind of lengthy here. Inhibitory neurotransmitters, different neurotransmitters that will inhibit. Glycine, there'll be a poison that blocks glycine receptors, which will produce death by asphyxiation. GABA is gamma-aminobutyric acid. It's one of the more common neurotransmitters in the brain, used by 1/3 of the brain neurons. Degeneration of this secreting neurons will result in Huntington's disease. So again, lots of different receptors. Basically what this chapter wants you to realize is that there's lots of different neurotransmitters that get released between neurons. And then there's an even more increase in the number of receptors that they can get bound to and to cause a channel or an interaction. And a lot of drugs, again, affect these different types of receptors. And what drug you're on will affect a receptor that will control if a neurotransmitter gets through to the next neuron or it doesn't get through. So it's really able to control. Neuropeptides, again, as another example of a neurotransmitter. Many chemicals used as hormones or paracrine signals are also found in the brain. CCK is involved in the feeling of satiety after a meal, so feeling full. Substance P mediates sensations of pain. Neuromodulators, it releases a classical neurotransmitter like acetylcholine. Endogenous opioids, these receptors were discovered to bind with drugs such as opium and morphine, resulting in pain relief. Opioids also produce euphoria, so they may mediate reward pathways. They may be related to feeling of well-being after exercise. So the opioid drug crisis, you can see how opioids are really craved after because they not only take away pain, they're given for extreme pain, opium and morphine, but they're related to kind of an exercise high or a euphoria high. Neuropeptide Y, this is the most abundant neuropeptide. It plays a role in your stress response. It's a stimulator of hunger. Leptin will actually inhibit this neuropeptide, so it will release to suppress appetite. So some people will take this if they're trying to eat less. Endocannabinoids, I'll let you guys read through some of these. Again, a lot of detail here. Marijuana is a type of this. Marijuana use will impair learning and memory because the action of THC is widespread and not actually controlled. So one of the downsides of marijuana, just that THC is very extremely widespread and very hard to control. So it will impair learning and memory. Nitric oxide is a gas. It will diffuse across presynaptic axons. It will diffuse into target cells and will activate these cyclic GMP second messengers. It causes blood vessel dilation and helps kill bacteria. So you're often given nitric oxide if you have had a heart attack, so it'll try to help to increase blood dilation in the heart. It's also responsible, the drug Viagra works by increasing nitric oxide release because it increases dilation in really all parts of the body. Carbon monoxide is another gas used as a neurotransmitter. It will activate the production of cyclic GMP in target cells. It's used in olfactory or your smelling epithelium in your nose, so carbon monoxide is there. ATP and adenosine as neurotransmitters. They'll be used as co-transmitters, so they'll be released via vesicles at the same time as a neurotransmitter. Chemicals that are or may be neurotransmitters. So these are even more a category and the chemical that could be a neurotransmitter. And again, a neurotransmitter is anything that will be released from the end of an axon in a neuron to travel across a synapse or a space to another nerve or axon. Synaptic integration. So this is kind of the last part. Hopefully you paused the PowerPoint or my video at some point to take a break. But these are neural pathways, and I want you to have a general understanding of neural pathways. They can be divergence or convergence. A divergence means axons have many collateral branches, so one presynaptic neuron can form synapses with several postsynaptic neurons. So that just means that one axon will diverge into many neurons that it's connected to, the postsynaptic. Convergent means several different presynaptic neurons, up to 1,000, which is a crazy amount, can synapse on one postsynaptic neuron. Summation means spatial summation occurs due to the convergence of signals onto a signal postsynaptic neuron, and temporal summation is due to a successive wave of neurotransmitter release. So this is a look at spatial summation. These are graded potentials, this EPSP. Synaptic plasticity is the repeated use of a neuronal pathway. It may strengthen or reduce the synaptic transmission. It's called a long-term potential when repeated stimulation enhances excitability. It can improve the efficacy, so the ability of it to perform its function. It's seen in learning and memory in the hippocampus part of your brain. Long-term depression occurs when glutamate-releasing presynaptic neurons stimulate the release of these endocannabinoids, and this suppresses the further release of the neurotransmitter. I'll let you guys read through this. Synaptic inhibition, more synaptic inhibition, presynaptic inhibition, again showing where it could be inhibited. And that's the end of this slideshow. I know this was a lot, guys, so check out those end of chapter questions, come to my office hours, think of big picture for this chapter, and I hope you guys are all healthy and well.